So I'm going to tell you all about my research today, which is I mostly started during my postdoc, and then I brought to UBC to start my own lab here just over a year ago. So a lot of what I'll be talking about today was work that I did in California, but I'm starting to integrate BC and Canadian ecosystems into what I'm doing as well. And you'll see that a lot of what I was studying in California is relevant here in addition to the rest of the world potentially. So yeah, let's get into it and talk about how fungi survive and thrive after fire, literally rising from the ashes. And I am really fascinated by this time period, almost immediately after fire. When the forest might look something like this, it can look pretty bleak from our perspective, our human above ground perspective. But a big part of what I want to talk about today is all of this life that is really vibrant at the soil surface and below the soil surface. Can be within days after fire, and I sort of span that year, first year after fire. Um, and I'd argue that these ecosystem dynamics that are happening within that first year following fire really lay the foundation for the rest of post-fire recovery. And so that's why I think this research is both really exciting and really important, because whatever these fungi are doing is going to impact the trajectory of recovery because they're there first. You might notice in this photo, there's also some lovely mosses, which I actually know nothing about, but I'm very keen to chat with some biologists if there are any moss people here. So yeah, my research focuses on the fungi, which we can see two of here. Can you guys see my cursor at all? No? Yes, I see nods. Okay, great. So you can see my cursor. I'm moving it over this colony of Pyronema right now, and there's another little fungus down here that's Geopixis. So yeah, I mean, just to reiterate what I just said, I think understanding how post-fire ecosystems function is really critical to supporting their recovery. So for my talk today, I'm going to talk about four things. First, I want to talk sort of abiotically about what fire does. And then I'm going to talk about one of my favorite fungi, Pyronema, that I affectionately call a first responder. And then I'll talk a little bit about some specific adaptations that fungi have to fire and what does it mean to be pyrophilous or fire-loving. And then I'll get more into the broader community of fungi that have this very dynamic response right after fire. So, from the perspective of soil, fire is kind of fire. It doesn't necessarily matter if it was an intentional fire or a wildfire. We see the soil fungi and other microbes in the soil responding to fire similarly, regardless of the kind of intention behind it. With more intense extreme fires, we do see a more intense extreme response. But there's similarities regardless of the type of fire. So you may have heard of wildfires and more intentional prescribed or cultural burns. I'm just showing examples of those here. So what does fire do to an environment from the perspective of fungi in that environment, and especially fungi that are inside of the soil? And so I like to keep this sort of cartoon of a general anatomy of soils in mind that I'm going to slowly reveal here. And so what I'm showing here is a soil profile. And so at the top, we have the soil surface where the fire happened, easily reaches an excess of 300 degrees Celsius, could be 1,000 degrees Celsius, like truly mind-boggling temperatures at the soil surface. This heat is enough energy to catalyze the formation of some pretty gnarly chemistry. So we collectively call this layer pyrolyzed organic material. You can just call it charcoal, is the vast majority of what this is. And all of these ring structures, we call these polyaromatic hydrocarbons, which is a phrase you may have heard before more in the context of environmental pollutants or carcinogens. But these are all naturally occurring compounds in post-fire landscapes that are the result of fire. And so that's another reason I think these fungi are really fascinating to study because they have evolved living immersed in this stuff. And I think we have a lot we could learn from them about how do we deal with these polyaromatic hydrocarbons. Um, okay, but first the fungi have to survive the fire. So let's continue diving down into this soil profile. And at some point, there is this layer where the heat penetrates, but not all the heat, in a range of around 60 to 100 degrees Celsius, is about the range that we would expect for things to not be able to survive. This is hot enough to kill anything that's alive in this area, but it's not hot enough to catalyze the formation of this really gnarly chemistry. And then diving deeper still, at some point, we're going to have little to no effect of the fire. And something that was really surprising to me early on in my postdoc work was we were going out sticking temperature probes in the soil under controlled burns that we were doing. And we realized that this can all happen within just a couple centimeters of the soil surface, depending on the intensity and duration of the fire. Um, but yeah, I think there's a lot of capacity for survival within the soil. So, I want to talk a little bit more about what is charcoal. This is our pyrolyzed organic matter in sort of its purest form. And there's this really fascinating whole entire other field of chemistry devoted to figuring out the structure of different charcoals because it's such a like complicated and fascinating structure. And these are some images from a review paper summarizing a lot of that work, where we have a handful of charcoal. We can zoom in with scanning electron microscopy, and it looks something like this in A, and then we can zoom in further with scanning electron or transmission electron microscopy, and it starts to look something like this, where we see these regions of disorder and then highly ordered regions. And so this is where the authors of this paper started to sort of extrapolate and hypothesize that perhaps these highly ordered regions that they're seeing might have a chemical structure that looks something like this down in the bottom corner in E, where it's just this solid wall of these aromatic carbon rings. It's a pure carbon wall, versus these more disordered regions might have a structure that looks a little bit more disordered, like in D here. But what I want you to take home from this is that it's a lot of these carbon ring structures. These are all polyaromatic hydrocarbons, is really what charcoal is. And the number of rings that are stuck together is directly proportional to the amount of heat that is applied to whatever the charcoal was originally, a tree or a plant of some sort, usually. And there was this hypothesis that this charcoal that, okay, so on the x-axis, we have increasing temperature. And then on the y-axis, we have a combination of the number of these aromatic rings and also how stable these authors thought they might be in the environment. So at some point, they're saying if you stick too many rings together, it's going to be inaccessible for any living organisms to break these open and access the carbon that's inside of them. It took a lot of energy to make these from the fire, and it's going to take a lot of energy to break them apart and access that carbon. So these authors were hypothesizing that it would be biologically impossible to break these open and access the carbon in them. Spoiler alert, I showed something different. So that was just carbon. Fire broadly dramatically alters the chemical and physical properties of soil. Some things that are important for life, it increases the pH. So if you've ever experienced indigestion, you know minute changes in pH can have dramatic consequences to life. Fungi included. Fire also makes the soil very hydrophobic, which means that water doesn't infiltrate down into the soil as easily. It's hydro means water, phobic means afraid of. So it's like the soil is literally afraid of water, which is how a lot of erosion can happen after fire as well. And there's this, I call it a nitrogen disequilibrium. I think there will be like these patches of nitrogen deserts, but then where you have areas of rich necromass, that layer where it got hot enough to kill everything in the soil, those are going to be areas that are like very enriched for nitrogen. And so organisms are going to have to navigate these nitrogen deserts and nitrogen jackpots that are sort of heterogeneous through the landscape. And speaking of heterogeneity, fire in general moves through a landscape in a very heterogeneous manner. It doesn't necessarily burn everything equally. Sometimes it'll jump over a spot, or there'll be a boulder or a fallen tree, or a wind pattern will change and it'll suddenly shift. And it's, yeah, fire's a dynamic force that creates a dynamic landscape that it leaves behind. So, with this idea of what a post-fire soil looks like in mind, how the heck do organisms, fungi in particular, make a living in this post-fire soil? And we were talking at the beginning about hunting for morels and all the other fungi that are fruiting like within a year after fire, and they're fruiting at the surface. They're having to contend with this layer of charcoal. And so how do they do that? It seems like a very inhospitable place, but these fungi also seem very happy there. So how do they do it? That's why I'm here today, and that's what drives my research. And to begin to tell this story, Pyronema has been a very central organism for me. So it's the genus Pyronema. This is what it looks like fruiting after a wildfire. It's an ascomycete, so it makes a cup-shaped fruiting body. Um, and there is a laurel leaf down here in the bottom corner. So these guys are very, very tiny, little teeny tiny cups that will fuse together into this kind of funky amorphous pink mat that we're looking at here. Um, and and we see it globally following fire. So this is just a screenshot that I took earlier today of current iNaturalist observations for Pyronema. There's currently 890 observations, and each one of these orange dots is somebody that's gone out into the environment after a fire. It could be a fire pit, it could be a wildfire, it could be a cultural burn, it could be a prescribed fire, um, could be a spot where somebody threw their cigarette and it burned a little bit. Um, but we only ever see it fruiting after fire. And I actually do go into iNaturalist periodically and validate all of these. So I'm reasonably confident that these are accurate, although I haven't done that in a couple months, so there might be a few outliers on here that are inaccurate. But the point is that Pyronema is responding to fire globally, regardless of ecosystem, location. I really think of post-fire ecosystems as sort of their own unique ecosystem that is neither the ecosystem that preceded it nor the ecosystem that it will become as succession progresses. And Pyronema, in addition to a lot of the other fungi I'm going to talk about today, are those sort of um, early colonizers. They're the first ones that are very active within days to weeks after fire. And this is an illustration of that activity. So on the x-axis is time since fire, with the fire sort of middle left-ish happening here. And so this is a summary of a lot of DNA sequencing data where DNA was extracted from soil and then amplified to target just the fungi that were there, and looking at all of the fungi that are there. And then we quantified out of all the fungi that are there, how abundant are they? And so this top row is Pyronema, and anywhere that you see a colored line on this plot means that we detected these genera. So I'm focusing on Pyronema right now, but there are several others. Right, I think I actually cover this up so we can focus a little bit better on Pyronema. And the darker that the line is, the more abundant it was, and like approaching 100%, which is pretty wild. The highest that we've ever seen it is like 60% of the whole community, which is bizarre. If we go outside right now and grab a handful of soil and sequence all the DNA in that soil, the most abundant, the dominant fungus in that soil might be like 5% of the entire community. So for Pyronema to make up 60% or even 40%, even 10% of all the fungi that are in the soil is pretty wild. So um, it's everywhere, it's dominating, it's really in this charcoal layer, just thriving and loving life. How does it do it? Some good news for me is that it's great to grow in the lab. It grows really easily and quickly. One of my collaborators and mentors who came before me isolated a bunch of these pyrophilous fungi and sequenced their genomes, which has enabled a lot of the work that I do now. So we use those sequenced genomes to enable doing RNA-seq. So this is looking at which genes are going to be turned on or activated when we grow Pyronema in charcoal versus a non-charcoal. And then, so this tells us, yeah, which genes are being turned on, but it doesn't necessarily tell us if those genes are functional, if they're doing necessarily what we even think they're doing. And so to more directly get at this question of, is Pyronema potentially eating the charcoal, which at the time the literature said it would be impossible. Like those, it takes a lot of energy to make those polyaromatic hydrocarbons, and it would take an unreasonable amount of energy for any reasonable organism like Pyronema to break those apart to access the carbon. Um, but we just decided why not? We'll just see what does Pyronema do if we give it a stable isotope labeled charcoal so that we're able to track the movement of the carbon that came from that charcoal because it's a different isotope than the rest of the carbon that is around us all the time. So we're able to track the movement of specifically carbon atoms that originated from this charcoal. And so I'll just jump to the punchline, which I've been teasing this whole time, is that Pyronema, in fact, eats the charcoal. And so on the right, our stable isotope experiment is telling us that this black square line on the bottom that's slowly increasing is saying, yes, Pyronema is taking charcoal and converting it into CO2. And I'll get to this other line in a second. And then this big complicated figure on the left that you really don't need to know the details of, this is showing the metabolism of polyaromatic hydrocarbons. And that every single gene that would be required to metabolize these aromatic rings, all of these genes are turned on, highly turned on. And so Pyronema possesses the ability to degrade these polyaromatic hydrocarbons, and it is doing it. This other line, so when we did this experiment, I worked with some collaborators who do these experiments all the time and have a million different controls to ensure that we're not detecting other sources of carbon, and we're really zeroing in on the carbon that was in the charcoal. And so despite accounting for several other potential sources of carbon in the system, this other line is telling us that Pyronema was able to find some carbon from somewhere else that we don't know where that was coming from. So this is kind of telling us that, okay, yes, Pyronema can eat charcoal, but it really doesn't like to. It will find things that we didn't even know were there and convert those into CO2 and metabolize that for carbon. I will tell you that we used the charcoal that we used for this experiment was made at 750 degrees Celsius, which is extremely hot. That is sort of at the limit of charcoal production, and that was sort of our pie in the sky. Like, if Pyronema can eat the most extreme charcoal possible, then it'd probably have a much easier time with charcoal that's produced at lower temperatures as well. And so we did a follow-up experiment where we actually took some charcoal produced at lower temperatures. So this is 350 degrees PYOM is charcoal, or 550 degrees charcoal, and we just added it to a whole soil microbial community. So it includes Pyronema, it includes all the other fungi that are there, it includes bacteria and like little insect larvae and everything that's in there. And when we give that complete soil community this stable isotope labeled charcoal, so we can track the movement of the carbon through this system, this top line is showing us that by far these post-fire soils are preferring to metabolize charcoal over on the bottom here near zero is where like organic carbon that's just easily naturally available is. And so not only are these fungi and other organisms living and surviving in these like very burned soils, they are really thriving and they are using that charcoal for a source of carbon, which I just think is totally wild. Um, so what about all those other microbes and all those other fungi beyond Pyronema? And to start to talk about them, I want to talk a little bit about adaptation and what does it mean to be truly adapted to fire. So we see several species of fungi consistently fruiting after fire. This is a friend and a colleague of mine holding a very charismatic little clump of high diversity of post-fire fire adapted fungi that it gives you a nice idea of how tiny these guys can be. Um, but yeah, so fire is triggering fruiting. That's an adaptation to fire. It's a response to fire. Um, and I would argue that both Pyronema and a similar looking fungus, Anthracobia, are very clearly pyrophilous. So these are fungi that we only ever see following fire. We never see them in a situation where there hasn't been some sort of intense amount of heat or fire itself. So, for example, a lot of these fungi also fruited after Mount St. Helens blew, which was a lot of heat. And sometimes you can see them on like very hot industrial sized compost piles as well, which is interesting. So it's not necessarily about the fire, it's maybe about the heat and the chemical changes induced by the heat. Okay, so what does it mean for a fungus to be pyrophilous? I like to differentiate between pyrophilous, true fire-loving fungi and simply fire-tolerant fungi. So when I'm talking about pyrophilous, fire-adapted fungi, these are fungi that are really unique and special to burned hot places or places that have previously been hot. Versus fire tolerant species are ones that we might see thriving after fire, but we also see them in other conditions. And they might be quite common in other conditions. But they might just take advantage of the fact that there's less competition in a recently burned landscape. I really like this example. This is from California. This is a mushroom that we call the redwood rooter that grows in the redwood forests in coastal California. And it's called the redwood rooter because it has this very long, it looks kind of like a tap root, although fungi don't technically have roots. Um, But this is a hint of how perhaps it might have been able to survive if it's very deep in the soil, and then it's able to fruit very rapidly post-fire. And we see these everywhere post-fire, but we also see them everywhere pre-fire too. So I don't tend to think of these as pyrophilous, but they certainly seem to be fire tolerant and taking advantage of a post-fire landscape. And there's a lot of fungi that will do this. But I get excited about the ones that are unique to fire, that are seem uniquely adapted to this burned landscape. What is that about? So I mentioned fruiting. The fruiting mushroom, that fruiting body, that's spore production. So the whole purpose of a fruiting body or a mushroom is to produce spores and get them out into the environment so that they can reproduce. And a lot of fungi are very clearly triggered to produce their spore producing structures as a response to fire. Some fungi require fire in order for their spores to germinate. So this is a photo of the fungus that I did my PhD on. It's called Neurospora crassa. It's a wonderful model organism, a total lab rat for studying genetics and molecular biology. And every day that we would work with it in the lab, first thing we had to do to work with the spores is we'd heat them to about 70 degrees Celsius, or you can plate them with liquid smoke like you would get at the grocery store. And that's what it needs to germinate. So that's an adaptation to fire. A lot of post-fire fungi make this funny little structure called a sclerotium. It's basically the fungal analog of a potato that's just this like amorphous, hardened blob of fungal hyphae. Morels are triggered to produce tons of sclerotia right after fire, and they're also triggered to make delicious mushrooms. Other species, their sclerotia are triggered to germinate. And so here you can see a sclerotium germinating and right away making a little mushroom here, which is pretty cute. So lots of adaptations, responses to fire. Um, and I sort of broadly think about the fungi that we see after fire as employing two different strategies, broad strategies. So they're either like investing in migration and reproduction and producing lots of fruiting bodies and lots of spores and getting their next generation out into the world, or they're really invested into making this post-fire habitat a home. And so I introduced Anthracobia and Pyronema earlier, and I think they nicely fit these two descriptions where we see tons of Anthracobia after fire. If you've ever been out in a recently burned area and seen these like carpets of cute little orange cups, it's a good chance that was Anthracobia. But when we sequence the DNA in the soil, it can be really difficult to detect Anthracobia. So it seems like it's just at the soil surface, making its fruiting bodies, making spores, getting out of there, and it's not really invested in making that post-fire soil a home. Whereas Pyronema can be much more elusive to find its fruiting bodies. But when I mentioned earlier that when we sequence the DNA in the soil, sometimes it's making up over half of all the fungi we can detect in the soil. So Pyronema is really invested in this post-fire habitat, which is, again, why I think it's so exciting. Okay, I think I saw there's like a bajillion questions or things in the chat rather, and then that went away. Maybe I'll get to the questions at the end when I can find where that little doohickey went. Okay, I'm just gonna keep going. Um, okay, so I want to talk a little bit about the dynamic community response. So not just Pyronema, but all the other fungi. What are they doing together in the soil? What is really happening below that soil surface? So you can see kind of how Pyronema often grows after fire. It likes these very shaded little pockets, which is part of why it can be tricky to find. So Yeah, I mentioned fire slicks for highly dynamic pyrophilous fungi. So this is some soil sequencing data that I'm showing you a little snapshot of. And on the top, I'm showing the abundance of Pyronema, and on the bottom is the abundance of Geopixus, which looks like this. And this was a big field study that we did where we did prescribed burns in two different sites, and then we had two other sites that we didn't burn. And so this is showing the abundance of these two fungi over time in those four sites. The sites that we burned are in orange and red. The sites that we didn't burn are in green and blue. And what I want you to take away from this is that Pyronema was very responsive. To the fire. GeoPixis was also, but it really differed between our two plots. Um, uh, and like Pyronema and GeoPixis were totally different. It just sort of felt a little bit like chaos looking at these data of like, okay, they're doing stuff. What are they doing? And so this is where I went into, yeah, what are they doing? Here's the one photo of a morel in this presentation. And so this is where I went into the lab. This is what morels look like when we grow them in the lab, just their hyphae. And we have this wonderful collection of these fungi that we can grow stably in the lab. This is what they look like when we're growing them in the lab. And we have genome sequences for all of them, which I think is going to enable some cool stuff coming soon. But for right now, I just wanted to play with these in the lab and try to understand how they're interacting together as a community in sort of the laboratory context. And so I think about this as either direct interactions or indirect interactions are sort of the two main ways that two organisms can interact if they're interacting, which these likely are. So this is what direct interactions look like in the lab, where we put two different fungi on the plate. So we inoculated Pholiota over here and Geopixis on this side. And this zone, we call this a zone of inhibition, where Geopixis is secreting something that is inhibiting the growth of Pholiota. So this is a great way to identify negative interactions where Geopixis on the bottom here might be doing something to exclude Pholiota from its habitat. Um, and so I did this in all possible pairwise combinations. And somewhat surprisingly, the vast majority of these fungi inhibited the growth of Pyronema, is what these flat arrows mean, that growth was inhibited. So Lyophilum, Pholiota, Coprinellus, Geopixis all inhibited Pyronema. And then a couple of these had other interactions as well, which might seem strange given how abundant Pyronema is right after fire. Um, so maybe they're not interacting directly. What if oh, yes, this could be an indication of some sort of chemical warfare going on. Um, but yeah, what if they're not interacting directly? What if they have a more indirect interaction? And so I developed this method for studying indirect interactions in the lab where this is one of those Petri dish plates that we're looking at from the side that is made of just charcoal, and agar is what we use to make it like a surface, kind of like Jello. And then we put a cellophane membrane on top and inoculate with Pyronema on top of that. Let Pyronema grow. Since we always see Pyronema first, we just started with Pyronema. And then once Pyronema covers the plate, we can take Pyronema off and then put down a fresh new membrane and inoculate with a second fungus. And so now this second fungus has to deal with the legacy that Pyronema left behind in this charcoal environment. And then we let it grow, take it off, put on a third one. And now this third fungus has to deal with the legacy that was left behind by whoever fungus number two was and Pyronema. Um, so after doing all possible combinations of this, in this fungus number two position, the majority of these guys were actually inhibited by Pyronema, which contradicts everybody inhibiting Pyronema when it's direct interaction. So Yeah, dynamic interactions. And then in this fungus number three position, this gets much more complicated. And so this network of interactions, it has been sort of the foundation for my lab. And okay, I just saw the chat thingy pop up really quick. I'm going to take a quick look and make sure there's no emergency. Okay, I see some great questions, but I'm almost to the end, so I'm just gonna go for it. So, my first question after all this, right? So, oh my goodness, whatever. That went too fast. Yeah. So in this beginning, everybody hates growing after pyronema. So why does everybody hate growing after pyronema? And I had a hunch it'd have to do with nutrients. So I just added nutrients back to this pyronema conditioned or pyronema influenced charcoal environment. And so this is the data showing that, where if we add back carbon, nitrogen, phosphorus, and iron, we see a restoration of growth. So just those four compounds is enough, and actually just nitrogen is enough on its own to restore the growth of trichorina growing after pyronema. And for all these other fungi, it's a different story, but it seems like pyronema possibly is really good at depleting its environment for nutrients. And so then that in turn inhibits the growth of the fungi that follow it. But if these fungi directly interact pyronema, they have some sort of chemical warfare shenanigans that is going to inhibit the growth of pyronema. So it's dynamic. Yeah, even more interactions. Okay, so that was a lot. I'm going to summarize everything that I talked about really quickly, and how fire transforms the chemical landscape of soil, generating this rich layer of polyaromatic hydrocarbons. And the microbes and fungi that inhabit this soil are really well adapted to metabolizing this altered chemistry and living with that altered chemistry. And fire selects for pyrophilous fungi specifically, especially pyronema. And pyrophilous fungi are super dynamic, which is really interesting and fun. I think there's a lot of direct competition and chemical warfare happening and indirect competition for nutrients. But if you take nothing else away from this presentation, I hope you now have an appreciation for the fact that post-fire ecosystems are really resilient and full of life. And I just think that's super cool. So tons of people to thank from my postdoc and graduate work at Berkeley. Thea Whitman is my stable isotope charcoal collaborator who was formerly at University of Wisconsin, and she just moved here to UBC. So I'm super stoked about that. And my growing baby lab at UBC, the Fisher Fire Fungi Lab. So with that, I would love to take some questions if I still have time.
Oh
Thanks,
yes,
everyone.
there's plenty of time. So you found the chat. Do you want to go through the questions in the chat?
Yes. Yeah, I found the chat. Okay, I'm scrolling up to the top so I can do this in chronological order. Um, yes, interactive chats. Oh, I'm sorry, my cursor was small. Um, Okay, I suspect that water immediately following a fire of great intensity is absent, and yet pyronema is able to grow. How is that possible? Fantastic question. Yes, pyronema and all of these fungi need water to grow. And so when we do see them within days after fire, it's because there was some sort of water event. It could have been water used to put out the fire. It could have been fog drip. It could be a seep, a stream, something. Or it could be after snow melt also. So yeah, they need fire and they need water. Great question. Um, how might pyronema, yeah, okay, how might pyronema affect how plant material shows up in the fossil record, like in carbonized plant remains in the Nanaimo group or local Eocene sediments? Yeah. So, oh, Azmarat Berhe at University of California, Merced, who's a soil scientist, told me. So she looks at like fossilized charcoal and whatnot in the soil, and she told me that the fossil record um, of charcoal, there is a fossil record of charcoal, which is part of why there is this hypothesis that biological organisms wouldn't degrade charcoal because it does persist in the biological record, but the quantity of it is far reduced from what we would expect if there was no metabolism of charcoal. So I feel like my research and now others is sort of filling this gap of, okay, so what happened to that charcoal deficit that's in the geologic records? But I think there is some proportion of it that doesn't get metabolized, and that's what persists. I don't know about the Nanaimo group or local Eocene sediments specifically, but I'll have to look that up. That was from Jennifer G. Thank you. Okay. Jade Hopkins says, Dr. Fisher, I was curious if you have been following research about the impact of wildfire smoke and fungal infections in patients. I have not. I am intrigued. It made me curious if fires also spread other types of fungal spores to other regions after big wildfires. Oh, okay. Thank you for the link. This is slightly outside of my um what I'm usually looking at. So yeah, no, I wasn't aware. I know there is Lita Kobziar, who I believe she's at University of Idaho, has been studying the fungal spores or fungi that are in smoke. I'm not sure how viable they are, but it seems like smoke could be a vector for dispersal. Just, I mean, smoke is sort of like wind that we can see in a sense. And we know fungal spores are in the air around us all the time right now. We're breathing them in. They're flying around in air columns with or without smoke. But smoke might enrich for certain ones. But certainly, I mean, smoke is hugely detrimental to our health and our immune system, and that can create an opportunistic situation for fungal pathogens. A lot of human fungal pathogens are very opportunistic. A lot of them attack the lungs, so I can imagine how that would be. No bueno, not good. Are those sequoia forests? Yes, redwoods are a type of sequoia, Sequoia sempervirens. Um, oh, I just answered the question. Great. Nice IDing sequoia leaves. Is that cause Carol Tran says, does it cause some diseases for plants? So all of the fungi that I have shown today do not cause plant disease. They are decomposers. They aren't particularly associated with plants one way or another, except for decomposing very dead and burned plants. Although we do sometimes see an increase in plant pathogens post-fire, and I think that's part of that um, fire tolerant story that they're not necessarily fire adapted or pyrophilous, but a lot of pathogens are opportunistic and they will take advantage of a lack of competition and fresh, tender new plants emerging to attack them. So, um, yeah, dynamic ecosystem. Okay, Christine's iPhone says, should pyrophilous fungi purposefully be spread over burnt forest to speed up recovery rather than planting trees right away? Yeah, great question. So I always caution moving organisms around. This is how some of the most classic examples of invasive species happen, is that we move one organism from one area to another where we think it's going to be a benefit, and then there's some unintended consequence. I suspect that we might be able to enrich for them by like doing things like adding nutrients and water somehow. Um, it's also encouraging to see that they kind of seem to be everywhere. And my current grad student is actually looking at the spore bank in BC soils, seeing if like how widespread are these fungi in the soil already. I suspect that they're already there and they're just waiting for a fire to come through. But doing things like lower intensity controlled burns can help sort of enrich the soil and sort of inoculate it. So it's building up that source of inoculum by doing more controlled burns. And so then if a higher intensity fire comes through, that inoculum is already there. It's already been seeded. So I think we can just encourage what's already happening is better than transporting things from elsewhere. Okay, Talia, where do fungi come from after fire? I think I just answered that. Does part of them survive or do spores arrive after the fire? Yeah, I think they're surviving in the soil. David says, if charcoal is used as a soil supplement, should the temperature of combustion production be limited to produce an optimum fertilizer? Yeah, great question. I think it depends what your goals are. So some of the biochar um, oh my goodness. Yeah, another benefit of adding charcoal is that it's a chelator, meaning it will sort of like pull things out of the soil like heavy metals or other toxins potentially, and they'll like stick to the charcoal, and so maybe less likely that those things will end up in your veggies or whatever you're growing. Um, but if you want it for the carbon aspect of it, then yeah, lower level combustion would be more degradable for sure. Um, okay, another one from Carol Tran. So is fire good or not so good? If it is good, then should we burn the forest once in a while? I believe the indigenous people still practice that and then get burned due to potential fire hazard spreading or more. Yeah, I mean, I think fire. Oh, should we even be talking about it in terms of good or bad? I think it's like spring cleaning. It's like I think it is ultimately good. Um, but it is easy for it to get out of hand for sure. And I think it is a delicate process and I mean, humans, we evolved with fire. Our digestive tract is the way it is because we learned how to cook our food over fire, and we've managed our land with fire for millennia until relatively recently, which I think you're alluding to the fact that, yeah, indigenous people still practice um, using fire as a management strategy. And I am definitely a huge proponent of more fire on the landscape, more good fire on the landscape. There's been some great research showing that like using fire to sort of thin out the understory can also help protect forests from more catastrophic fires. So yeah, I think there's a multitude of reasons that I think we should be burning our landscapes more in a controlled way. You're welcome. Thank you all. This is awesome.
So I have a question. I'm still a bit confused. When you were showing that slide really near the beginning and you were showing the temperature at the top, which was could be 600 to
Mhm.
1,000 degrees and nothing living could survive. And then when it got down to below 100, I think you said some things could survive. So how far, if it's not spores, but the mycelium of the pyranoma
Mhm.
that's surviving, how far, because you said when it's found, it's really, you only find it really near the surface.
Mhm.
How is it moving from, if it has to survive down where the temperature didn't get over 100, how does it get up to the surface where the temperature was at a temperature that would kill all the living things?
Yeah, great question. I mean, so this whole entire image could be happening within 2 centimeters. So I don't know how surface you're thinking, but if it's just 2 centimeters down into the soil, that's enough in a lot of places, in a lot of fires. It could also be conveniently under a rock or a spot where the fire just happened to jump over it. Um, so yeah. They might also be deeper in the soil. So those paranema grows very fast and very prolifically. So it might burrow down into the soil, um, which, yeah, I don't know to what extent it does that, but
And the other question I have is in terms of in the areas where you see pyronema and some of the other pyrophilic pyrophilic fungi. Um,
Mhm.
is there any like going back and checking to see whether those areas have more plant growth afterwards of um,
Yeah, so I've actually been writing a grant to test exactly that. That is my hunch. That's my hypothesis is that if you have these fungi growing, like knitting the soil together, helping water infiltrate down into the soil, they're like jump starting nutrient cycling. That seems like an environment that would be more amenable to plants germinating and plant success. But we haven't actually directly tested that yet. So stay tuned.
and can you show the slide again that showed like how long after a fire, how soon after a fire we'd see pyronema and how long it would last for? Just
Oh,
for
yeah.
if we're going out looking places where there's been fires.
Yeah. So the soonest I've seen it was 4 days after a fire. And that was in a friend's fire pit in his backyard, where they get a lot of fog. Um, so yeah, I think most wildfire areas you'd need very special permission to get in that soon.
Mhm.
There's also Neurospora, that fruits on trees. We can detect it in the soil, but it's a mold. It makes like pink, fluffy moldiness on very burned trees. And it does that like within days after fire. So I still have yet to see it in the wild. Because that one's very short-lived. But Pyronema and the rest of these guys will stick around for a bit longer. And if you're looking for morels, it can be a good strategy to follow the trail of these cute little cups, and then they might lead you to the morels. I usually get stuck with the cute little cups though and never make it to the morels.
So that would often be like maybe like even like 6 months or more later than the next spring after
Yeah.
you might still see pyronema. Like
Yeah, you can actually still see it for a couple years, but it declines in abundance and it can be trickier to find.
and
Uh,
what was the name of the other orange little cup one that you said was quite common?
yeah, that was Anthracobia. Let me.
And that you said was a bit easier to see. And does it also stick around like for months or
No, Anthracobia and Pyronema have a similar timeline, but Anthracobia is often like on the soil surface and it'll sort of form these mats. That almost reminds me of like an orange shag rug or something. It's very striking on the post-fire landscape where it's just black and then this like bright orange mat. Um, okay, there's some more questions in the chat from Talia. Does Paranema actually gain anything or need anything from burnt soil, or does it just have less competition? Yeah, it's gaining all of its nutrients from that charcoal, and from the surrounding soil. So yeah, it's really um, living life in that soil, eating what's there. A lot of it is the charcoal. Um, thank you, Nina. Okay, great questions, everyone.
Well, that was a great presentation. Thank you, Monica.